My name is Brian Kwan, and today I'll be presenting on SARS-CoV-2 variants. Since the beginning of the pandemic, there has been over 300 documented genetic variants of the SARS-CoV-2 virus. Recent studies seem to indicate that the SARS-CoV-2 virus that is causing this ongoing pandemic can be traced to a species of bats, as over 96% of its homology can be matched to that of coronavirus found in bat species in Yunnan, China. In recent months, four primary variants are of particular concern for public health officials. These four variants are designated as the alpha, beta, gamma, and as many of us have probably been hearing about this in the news in recent months, the Delta variant. All variants are of great concern for those with increased transmissibility and virulence. So variants of viruses also exist in other past examples of infectious diseases, such as ongoing infectious diseases such as the influenza virus. So the influenza virus has different types, and type A in particular is most often talked about as it often causes the seasonal flu. Now, due to new variants of influenza emerging in the population and infecting people, this does lead to ongoing sort of endemic like seasonal endemic disease, which we call the seasonal flu. The H1N1 swine flu or the Spanish flu of 1918 are examples of particular strains and particular variants that caused global outbreaks. As we first discuss variants of the SARS-CoV-2, I think it would be prudent to first discuss how these variants come about. After all, as we usually refer to pathogens that cause COVID-19 with the general term coronavirus, but coronavirus is a term that really encapsulates a multitude of different pathogen variants. So all of human CoV viruses originate from some animal source, and it is bats that in particular that make up a large reservoir of beta and alpha CoV viruses. So RNA viruses, such as the SARS-CoV-2, have a very, very high rate of mutation, certainly higher than other DNA viruses or even their hosts. And the reason for this is perhaps due to the lack or ineffective proofreading capabilities of the RNA polymerase. However, due to the large size of RNA genetic material, such as that found in the SARS-CoV-2, the mutation rate for this particular virus is around 0.8 to 2.38 times 10 to the negative 3 nucleotide substitutions per site per year, which is lower than most RNA viruses. And as nucleotide sequences can mutate, the gene expression may also change as a result due to mutated protein structures that could either increase or decrease the transmissibility of the virulence. From a perspective of natural selection, those mutations that are beneficial to the propagation of SARS-CoV-2 would most likely be preserved in the population, leading to variants of increased transmissibility or virulence. Or increased transmissibility. So most of the major changes to the RNA structure of SARS-CoV-2 is in the coding sequences for spike proteins. Spike proteins are these glycoprotein structures that allow for the virus to enter into the host cells. These spike receptors are able to recognize and bind to the protein receptors of a host cell and facilitate the fusing of the viral envelope with the host cell's membrane. Now, for the first variant we will discuss is the alpha variant, also known as B.1.1.7 lineage. In late December of 2020, reported cases of alpha variant SARS-CoV-2 have been documented in the United Kingdom. This particular variant contains 17 different mutations to its viral genome. 8 of these 17 mutations are found in the gene sequence that codes for the spike proteins of the SARS-CoV-2. The reason for the increase in transmissibility in this alpha variant is due to the mutations of the spike protein that increases the affinity of the virus to the ACE2 receptors on the outer membrane of the host cell. Because this increase in transmissibility, because of this increase in transmissibility, with reports indicating a 43 to 80% increase, the alpha variant easily becomes the most dominant and prominent variant found in the UK during this time. In one report, there is also an increase in rate of death with this alpha variant, with a hazard ratio of 1.67 as opposed to 1. And that's with a confidence interval of 95% compared to the non-VOC or non-variant of concern cases. Now, the next variant that we'll be discussing is the beta variant, or the B.1.351 lineage. This variant is responsible for the second wave of COVID infections to hit South Africa during the October of 2020. This variant contains 9 mutations of the S gene that codes with the spike protein, and of these 9 mutations, 3 are specifically associated with the receptor binding domain of the spike protein, of course. This increased the spike protein and the virus's affinity for the ACE receptors on the plasma membrane cell, allowing for greater transmissibility because it easily penetrates and fuses into the host cell. And yeah, thank you for listening to my presentation.